All right, let's dive right in. Today, we're taking a journey to the very heart of the cell, to its command center. We're going inside the nucleus, the incredible organelle that holds the blueprint for life itself. So, here's our plan for this deep dive. First, we'll get a handle on the nucleus's main job as the cell's command center. Then, we'll check out the amazing fortress that protects it. After that, we'll look at the exclusive gateways that control all the traffic, the VIP access system that manages it, and finally, we'll take a peek inside the factory running at its core. Okay, let's start with the big question. What exactly is the nucleus, and why is this tiny little structure so absolutely critical for pretty much all complex life on our planet? Well, the nucleus is really the one thing that separates complex cells like ours, eukaryotes, from simpler ones like bacteria. See, in bacteria, the DNA just kind of floats around in the cell, and reading a gene to build a protein happens all at once in the same place. But in our cells, the nucleus creates this private, secure office for the DNA. And that separation is a total game changer. By keeping the genetic blueprints separate from the factory floor of the cytoplasm, the cell gets way more control over which genes get used and when. That control is what made complex multicellular life possible. So, to wrap this idea up, the nucleus you're seeing here is both the cell's super secure library and its main control tower. Tucked away inside this double-walled envelope is the entire genome, the complete genetic blueprint. And from in there, it calls the shots for almost everything the cell does, from how it gets energy, to when it divides, to what it passes on to the next generation. Now, if you've got a command center that important, you've got to protect it, right? So let's explore the incredible double-layered fortress that stands guard over the cell's most precious asset, its DNA. Take a close look at this diagram. The nuclear envelope isn't just one wall. It's two separate membranes, one inside the other. And what's really cool is that the outer membrane is actually connected to another structure called the endoplasmic reticulum, or ER. It's basically an extension of it, and it's even covered with the same little protein-making factories called ribosomes. The inner membrane, though, that's its own thing. It faces the inside of the nucleus and has its own special set of proteins. And right underneath that inner membrane, there's this strong, fibrous meshwork you can see here called the nuclear lamina. It's made of proteins called lamins, and you can think of it like the steel rebar in concrete. It's the structural scaffolding that gives the nucleus its round shape and keeps it stable. So what happens if that cellular rebar, that structural support, fails? Well, this is where we see a direct and critical link to human disease. Just like what's shown here, if there's a mutation in the gene that makes these lamin proteins, the whole nuclear envelope can get weak and unstable. This failure has some really serious consequences, leading to a whole class of diseases called laminopathies, which include certain types of muscular dystrophy. It's a pretty stark reminder of how one tiny molecular problem can have a massive impact on the whole organism. Okay, so if the nucleus is this high-security fortress, how does anything get in or out? It can't be totally sealed off. Well, it turns out there are these massive, incredibly intricate gateways. And these gateways are the nuclear pore complexes, or NPCs, and they are just phenomenal molecular machines. On the left, you can actually see their 8-sided shape in a real microscope image. And on the right, the 3D model gives you a sense of just how unbelievably complex they are. These structures are the only way in or out. All traffic between the nucleus and the cytoplasm has to go through them. And to give you a sense of the sheer scale we're talking about, a single one of these pores has a mass of about 125 million Daltons. That's roughly 40 times bigger than a ribosome, making it one of the absolute largest protein structures in the entire cell. It's a monster. And this massive complex is built from hundreds, sometimes up to 1,000 individual proteins called nucleoporins, all arranged with that perfect 8-fold symmetry we just saw. This whole structure is perfectly designed for its job. On the outside, facing the cytoplasm, it has these long filaments that stick out like tentacles, sensing and grabbing onto cargo. In the middle is the main channel where things pass through. And on the nuclear side, there's a structure called the nuclear basket that helps receive the molecules as they come through. So we've seen the gateways. Now let's talk about the rules of entry. How does the NPC act as the ultimate border control checkpoint for the cell? So how does this border control work? Well, it's actually got two different modes. If a molecule is really small, it can just drift right through the channel, no problem. But for the big, important cargo, and that's most proteins and RNAs, it's a whole different story. They can't just wander in. They need a special VIP pass and an official escort to get through. And those official escorts belong to a family of transport proteins called karyopherins. As you can see here, they come in two main flavors. You've got the importins, which handle all the traffic going into the nucleus, and the exportins, which manage everything that's heading out. So how does import work? Let's follow a package. A protein that needs to get into the nucleus has a special address label on it called a nuclear localization signal, or NLS. An important protein sees this tag out in the cytoplasm and grabs onto it. This new complex then gets guided right through the nuclear pore. Once it's inside, a little molecule called rRNA binds to the important, which makes it let go of its cargo. The important then heads back out to the cytoplasm, ready to pick up another package. Now, export is a very similar process, just in reverse, with one key twist. For something to get out, it needs a nuclear export signal, or NES. Inside the nucleus, an exportin, the cargo, and rRNA all have to team up to form a little package. Then, that three-part complex moves out through the pore. Once it hits the cytoplasm, the rRNA gets switched off, which causes the whole package to fall apart, releasing the cargo. It's this difference, lots of rRNA inside the nucleus and not much outside, that gives the whole transport system its direction. All right, for our final stop, we're venturing even deeper inside the nucleus, to a dense, bustling area that has a truly massive job, the nucleolus. The nucleolus isn't just some random blob, it's a highly organized ribosome factory. It doesn't even have its own membrane. The reason it looks so dense and dark in these diagrams is because it's just absolutely packed with all the raw materials and machinery for constant manufacturing. This is the assembly line. The genes that code for ribosomal RNA or rRNA are read here. At the exact same time, ribosomal proteins are being built out in the cytoplasm and then shipped into the nucleolus. Yep, using that very same import system we just talked about. Here, the rRNA and the proteins are put together to make the large and small pieces of a ribosome. And the scale of production here is just mind-blowing. An actively growing cell has to crank out about 10 million new ribosomes every single time it divides. All of this is organized inside the nucleolus before the finished parts are shipped back out to the cytoplasm to start their critical job of building all the cell's proteins. So, we've seen that the nucleus is this highly regulated command center, a fortress with some seriously sophisticated border control. But that leaves us with a fascinating final question. If it's so well defended, how have invaders like viruses figured out how to sneak past security, hijack the whole system, and use the cell's own machinery to replicate themselves?